In this video, we are going to discuss regarding nitrogen fixation by blue-green algae, that is BGA. The blue-green algae or cyanobacteria are very special type of prokaryotes widely distributed all over the earth and that can fix atmospheric nitrogen through symbiotic as well as non-symbiotic methods. See, blue-green algae can fix N2 in aquatic as well as terrestrial ecosystems. They contribute very high amount of fixed nitrogen in agriculture because of their nitrogen fixing ability. They are used as a biofertilizers. Many crops like rice, banana, vegetables, etc. are benefited by these types of blue-green algae biofertilizers. Now, nitrogen fixation by blue-green algae through symbiotic method. The cyanobacteria have some special features and structure like heterocyst for nitrogen fixation. All these blue-green algae are having a special type of cell which is called as a heterocyst, which is specially responsible for nitrogen fixation. How? That we will discuss in the next slide. See, unlike rhizobia, they have unmatched host range such as fungi, lichen, bryophytes, pteridophytes, angiosperm and gymnosperms. All kind of plants, while cryptogams or phenograms are used as a host for nitrogen fixation by these blue-green algae. These are not restricted to roots only. These blue-green algae not only fix atmospheric nitrogen in root of the plant, but also they exhibit or they may be present in a thallus of lichens, thallus of anthoceros, leaf pocket of azolla or choroidal roots of cycas or and in angiosperm plant stem also. So the blue-green algae partner fix atmosphere nitrogen and supplies to host inhabited, while the host provides shelter to blue-green algae. The specialized structure of cyanobacteria having nitrogenase enzyme in it to fix nitrogen and a unique method or mechanism to protect nitrogenase from oxygen is called as heterocyst. So let's discuss regarding structure and function of heterocyst of blue-green algae. So this is the structure of heterocyst. Look at the here. This is the structure of heterocyst. These are the vegetative cells. Now heterocyst, a different but specialized cell in the thallus of cyanobacteria, which is highly thickened by many layers of cell walls. That's why it is called as a cyst, CYST. So look at here. It is the thick wall. These are the thin walled vegetative cell, while this is the thick walled heterocyst. The number, size and position of heterocysts differ in different species of blue-green algae. It may present at basal region, terminal region or intercalary in position. Look at here. Here is the intercalary position. Here is the basal position of heterocyst and here it is a terminal in position. In short, heterocyst is a large, transparent, thick-walled cell in some members of filamentous blue-green algae. The heterocyst is different from vegetative cell engaged in nitrogen fixation. Heterocyst is different from vegetative cell because it is engaged in a nitrogen fixation. As it contains, this heterocyst contains enzyme nitrogenase, thick walls, and this enzyme nitrogenase is protected from oxygen by thick wall of this heterocyst. There is a no diffusion of O2 from outside. Look at here. This structure. This is the structure of functioning of heterocyst. This is the heterocyst and this is the vegetative cell. This thick walled cell is called as heterocyst, and there is, due to this thick wall, there is a no diffusion of O2 from outside. At the same time, there is a no PS2. Here PS2 is inactivated, so that PS2 is responsible for generation of oxygen, but in heterocyst PS2 is inactivated, so there is a no evolution of oxygen. And in addition, helps to protect O2 sensitive nitrogenase enzyme. So due to thick wall and due to PS system 2 inactivation, there is a no formation or there is a no presence of oxygen, so that this heterocyst protects the nitrogenase enzyme easily. And however, the vegetative cell containing chlorophyll have PS1. This vegetative cell near to the heterocyst have photosystem 1, which is concerned with production of ATPs during photosynthesis. And this is very unique mechanism in blue-green algae, producing ATPs but not evolving oxygen, which can damage nitrogenase enzyme. So heterocysts are connected to adjacent cells through plasmodesmata to get nutrient. So whatever nutrient needed to this heterocyst is getting from the vegetative cells. And in this way, by avoiding oxygen presence, the heterocyst, with the help of thick walls and inactivation of photosystem 2, this heterocyst protect the nitrogenase enzyme and fix atmospheric nitrogen and provide this nitrogen to the host plant. Most nitrogen fixing cyanobacteria are filamentous, consisting of a chain of vegetative cell and heterocyst that function as nitrogen fixing machinery of the organism, and they form symbiotic association with fungi, lower vascular plants as well as angiosperm. So let's see different associations of or symbiotic nitrogen fixation of blue-green algae in different hosts. First, the symbiotic N2 fixation by blue-green algae or BZA in coralloid roots of the cycas. Look at here, these are the coralloid roots of cycas. These are the coralloid roots of cycas, and here inside this root there is a algal zone. In a cortical tissue there is a algal zone. These are nostoc, which are having association with the roots and fix the nitrogen by symbiotic process. The filament of cyanobacteria like nostoc infect the special type of apogeotrophic roots of cycas having many lenticels for respiration. The blue-green algae, when entered to coralloid roots, establishes endosymbiotic relationship. It started fixing atmospheric nitrogen in its heterocyst and provide the fixed nitrogen to host plant. So the nostoc filaments are present in the middle cortex here. You can easily see. Now next, that is symbiotic N2 fixation by blue-green algae in Anthoceros. Here the blue-green algae like Anabina is living endosymbiotically in the thallus of Anthoceros. Anthoceros is a bryophyte and fix atmospheric nitrogen in its heterocyst, which is having nitrogenase enzyme. The fixed nitrogen is supplied to Anthoceros, and in that it gets shelter in the thallus. So look at the diagram here. TS of thallus shows the presence of algae Anabina, which is having a large transparent heterocystic cells which fix the nitrogen and make it available to the Anthoceros. In return they get a shelter. Next, symbiotic nitrogen fixation by blue-green algae in aquatic fern Azolla. So this structure shows first one, that is entire leaf of Azolla, and B shows the Anabina in the leaf pocket of Azolla. The blue-green algae Anabina azole is a well-known endosymbiont in the leaf pocket of water fern Azolla. It fixes nitrogen in the leaf pocket which are available to different crops like rice after the death and decay of Azolla plants. Azolla is commonly used as a biofertilizer on large scale for paddy, that is rice, which increase the yield by 10 to 20%. So Azolla anabina in the leaf pocket of Azolla fix the nitrogen so that these Azolla plants are grown into the rice fields, and after death of these Azolla they provide a nitrogen to paddy rice or rice plants. Next one, symbiotic entomophyxation by blue-green algae in lichens. Now, some species of lichen contain cyanobacteria like nostoc in their thallus and perform nitrogen fixation symbiotically. Many times the lichen thallus show fungi, that is mycobiont, photosynthetic green algae, and nostoc, that is phycobiont. So nitrogen fixed by cyanobiont is utilized by lichen, and in return of that cyanobacteria get shelter in its thallus. But the detail of nitrogen fixation is obscure. Lichen thallus in tear shows upper cortex formed by fungal hyphae. This one. Below it there is a algal zone in which nostoc, the endobiont, is present. So this is the algal zone. It is then followed by medulla and lower cortex. This is the medulla and this is the lower cortex. Many times other than nostoc we get calothrix, oscillatoria, tolipothrix, etc. in lichen thallus as a cyanobacteria, that is algal partner. And these algal partners fix the nitrogen from atmosphere and make available to the lichens. So this is the symbiotic nitrogen fixation by blue-green algae in different kinds of host plant.